Understanding the structure and physiology of the eukaryotic cell through comparisons to the Petit Science Center. The Petit Science Center is a state-of-the-art facility that was built at Georgia State University in 2010. The eukaryotic cell is similar to the Petit Science Center at Georgia State University in that they both require many different pieces, each providing unique and essential services in order to function properly. Cell theory.
The cell theory states that cells are the basic unit of life. They can either be unicellular or multicellular. And it also states that cells arise from pre-existing cells.
The cell membrane.
The cell membrane is a lot like the walls of the science building. The membrane is a protective barrier that encloses the cell as well as helps it to maintain internal chemical conditions that differ from its environment.
The ability to maintain an internal environment is essential to the maintenance of cell life.
The membrane is composed of lipids. Each lipid has one end that is attracted to water and another which repels it. They face the inner and outer side of the cell membrane respectively. The membrane controls the transportation of materials in and out of the cell, and proteins in the membrane receive and transmit signals from other cells. In some cells, proteins in the membrane group together and carry out metabolic reactions. The membrane is dynamic. The proteins and lipids are not fixed, and the components are constantly changing.
Transport proteins.
Proteins also help transport small molecules through the cell membrane. These are called transport proteins, and they are similar to the security staff in the science building because they both control who is allowed inside. Membrane proteins move molecules in and out of cells through passive or active transport. Passive transport includes processes such as simple diffusion, osmosis, and carrier protein facilitated diffusion. There are two types of active transport, both of which require the cell to expend energy, often in the form of ATP.
Primary active transport.
Primary active transport uses the hydrolysis of ATP to move ions against their concentration gradients. An example of this is the movement of ions by the sodium-potassium pump.
A concentration gradient is a measure of how the concentration of a substance changes from one area to another. Secondary active transport.
Secondary active transport uses the energy produced by the electrochemical gradient created in primary active transport. In the sodium-potassium pump, the concentration gradient of Na+ supplies the energy required to transport glucose against its concentration gradient into the cell.
The nucleus.
The nucleus is the command center of the cell. It's a lot like the department chair of biology, Dr. Tai. The nucleus controls a significant portion of activity within a cell, including the production of proteins and the replication of DNA. The nucleus is surrounded by a double membrane, which controls what enters and leaves the nucleus.
This double membrane is called the nuclear envelope. Nuclear pores found in the envelope regulate transportation in and out of the nucleus. Chromosomes are composed of chromatin, which is made up of proteins and DNA, and are found inside the nucleus. The endoplasmic reticulum.
Now about the endoplasmic reticulum. There are two types. There's the rough ER and the smooth ER. The rough ER is rough because it contains ribosomes and are essential for protein synthesis. The smooth one is called smooth because it contains no ribosomes and is involved in lipid synthesis and detoxification. This network makes up 1/2 of all membranous tissue of the cell. The ER is just like the stairs and elevators of Petit Science Center because goods are manufactured and shipped to the needed areas, just like how the ER is a membrane protein transport.
The ribosomes.
Time to talk about the ribosomes. The ribosomes are used to create proteins needed for the cell's life by following instructions by the nucleus. They're made up of RNA and proteins, just like the electric room in the Petit Science Center.
Think of proteins as machinery for cell functions. Much like electricity is essential for the building, enzymes are a type of protein without which life could not exist. The Golgi apparatus.
Now we're going to talk about the Golgi apparatus. Proteins are sent to the Golgi apparatus from the rough ER. Proteins are attached to carbohydrates and lipids, then sent to their final destination. Like the Petit Science Center stock room, the Golgi apparatus is used for shipping those goods created by the ER and ribosomes to the rest of the cell.
The Golgi apparatus gathers simple molecules and combines them to make molecules that are more complex. It then takes those big molecules, packages them in vesicles, and either stores them for later use or sends them out into the cell. The mitochondria.
The mitochondria is considered the powerhouse of the cell. This is the site of cellular respiration and energy production. Here, chemical energy is converted into a more useful form for the cell. An example of this is ATP. In the Petit Science Center, the mitochondria is most similar to the building's generator and the electrical room. With both these items, energy is converted into usable forms and then outputted throughout the building in the forms of light and heat.
The mitochondria has both an outer and an inner membrane. The outer membrane covers the organelle, and the inner membrane consists of multiple folds called cristae. These folds increase the surface area and are the location of many of the chemical reactions that occur within the mitochondria. The chloroplasts.
Chloroplasts are the structures that produce food for the cell through a process known as photosynthesis. They use solar energy from the sun and convert this energy into sugars for the plant. The chloroplast contains two membranes, an inner membrane and an outer membrane, to protect the structure. Chloroplasts contain a stroma, a granum, and a thylakoid stack. The granum is made up of one thylakoid stack, and the stroma is the area of the chloroplast where sugars are created. The thylakoids have chlorophyll on their surfaces, and this chlorophyll is what creates sugars with the use of sunlight. The skeleton of the chloroplast is the stromal lamellae. The stromal lamellae connects the thylakoid stacks.
The Petit Science Center does not have solar panels, but if it did, we would compare them to chloroplasts. Solar panels take in energy from the sun and convert it to electrical energy. Chloroplasts function similarly in that they convert solar energy into chemical energy. The cytoskeleton.
Now for the cytoskeleton. The cytoskeleton has many functions to a cell. These include supporting the cell and maintaining its shape, holding organelles in position within the cell. It's involved in cytoplasmic streaming, which is movement of the cytoplasm, and interacting with extracellular structures to help anchor the cell in place. This is like the Petit Science Center's external framework and internal framework for the building. Just like the cell, there are different components of the framework. For the cell, there are microfilaments, intermediate filaments, and microtubules. Microfilaments help the entire cell or parts of the cell move. They also determine the shape of the cell and help to stabilize it.
Microfilaments consist mainly of actin, which is long and thin in composition. Actin is a protein that is found mainly in muscle tissues.
Intermediate filaments mainly aid in structural function. They anchor cell structures in place and resist tension by maintaining rigidity in surface tissues.
Intermediate filaments are not as thin as microfilaments, but they are not as thick as microtubules. This portion of the cytoskeleton is like a web of connections that provide stability to the cell.
Microtubules are the largest of all and are long, hollow, unbranched cylinders. They function in that they form a rigid internal skeleton for some cells. They also act as a framework along which motor proteins can move structures within the cell. The Petit Science Center is composed of cement columns that function to hold the building upright and sturdy. It also contains secondary columns and branching metal frameworks that aid in balance and shape. These properties are very similar to that of the cytoskeleton of a cell.
The lysosomes.
Lysosomes are membrane-enclosed organelles originating from the Golgi apparatus.
Enzyme proteins are created in the rough ER, packaged in vesicles, and sent to the Golgi apparatus. The Golgi finishes creating digestive enzymes and pinches off small, specific vesicles. These vesicles are lysosomes, which then float in the cytoplasm until they are needed.
They contain digestive sacs that can break down micromolecules in the cell using hydrolysis.
Lysosomes break down cellular components and ingested molecules that are no longer needed by the cell using lysosomal enzymes.
Like the waste disposal in Petit Science Center, the lysosomes prevent buildup of excessive macromolecules.
This project was created by Cynthia Carter, Latara Northcutt, and Shoshana Robbins. The diagrams were taken from Sadava's Life, 9th edition. Photographs of Petit Science Center were taken from the Georgia State website.